Okay, good morning, everybody. It's time to start. Welcome, nice to see you. First of all, please switch off your phones. Vibration mode. Okay. The SAME-NeuroID seminar series will be also broadcasted to Paris Brain Institute, Max Planck Psychiatry in Munich, and Erasmus Rotterdam. So this short, let me welcome Witek Konopka, who.
Thank you very much. I have two things to say. First is a short biography of Ali, our guest today, and the second important thing you will see. So Ali completed his medical studies in Pakistan, in Karachi, and then he moved to United States for his clinical training in Houston. Afterwards, he did, I wanted to say back to Europe, but not back in Europe, at the University of Zurich, and at ETH he did MD, PhD. So he's a physician and scientist, which is a very good combination for his science and for us today. And after time spent in Switzerland, he finally landed in Warsaw, and now he heads the Trend Lab at the BRAINCITY Center of Excellence, which is located at the Nensky Institute in Warsaw. And now it's time for the second thing about the lecture that we will hear today. I think it is important to mention that this kind of results and this kind of stories you will not see in the textbooks and school. And I would like to say even more that sometimes it is contradictory to everything what we have learned on the biology lessons at school. So I would like to welcome Ali Jawaid, and floor is yours.
Thank you very much, Witek, for this very, very kind introduction. And it's my great pleasure to be here and honor to be here. It's my second time in Port. Last time when I was there in 2021 at Port for Neuroscience, I remember my last statement was that this is the best conference I've attended. So, and it was truly meant. You know, I was not trying to flatter Witek with that. And I'm very hopeful that this experience would be as great as it was last time. Right. So let me start with lecture right over here. Well, a couple of years ago, when I gave a seminar like this, like, you know, as an independent group leader the first time, the title was a bit different. It's actually not metabolic basis of neuropsychiatric disorders, but more epigenetic basis of neuropsychiatric disorders. But what we have learned over the last, I'd say 3 years or so, is that metabolism and epigenetics, they go very much hand in hand, up to the point that now I actually would say that for the neuropsychiatric disorders, the single most important risk factor or prognostic factor, or even a mechanistic factor, is actually metabolism. And then there is something else which is very peculiar. It's many lives, one master. So that's actually a little tribute to one of these, like, you know, very fine pieces of, it's not really fiction, it's more like a fictionalized account from a psychiatrist, Brian Weiss over here, who described the story of one of his patients. A very interesting anecdote, because he had a patient, like, you know, this young lady who had inconsolable anxiety. And would have like crying spells, would have like, you know, these periods where she would like really detached from the reality and was in a lot of psychological pain. And then when he did all kind of exploration, there never seemed to be any kind of risk factor in her lifestyle or any of her recent exposures. They tried all different medications, none of these really worked. And eventually, Brian Weiss resorted to something more, you could say, non-traditional, non-conventional, and he tried hypnotherapy. And it turned out that while this patient did not have any traumatic experiences in her current life, there was a history of a lot of trauma in her past lives, And then during these hypnotherapy sessions, Brian Weiss actually tried to, heal the trauma from the past lives. So I know this is all kind of like a little bit of pseudoscience, right? I mean, this is not something which can be experimentally proven. This is something which perhaps doesn't have any implications for clinical psychiatry or, clinical neuroscience. However, this actually does allude to a very, very important concept, and that is that a lot of things which we experience, a lot of different susceptibilities which we acquire, they're actually not ours. They have been passed on to us from previous generations. So these are not our past lives, but actually, our ancestors which are transferring those. And over the course of the next 1 hour or so, I will explain how all of that is important, how experiences from our childhood are important not only for us, but also for the subsequent generations. I have a little video introduction to my lab, which I'll start with, and then we will build up on that from here on. Do you have sound for this? Or? Okay. Well, it doesn't matter. So it's a little bit about what we do in our lab, which is like a focus on neuropsychiatric disorders. At the mechanistic level, we look at metabolism and epigenetics, and we look at it in the context of environmental exposure, such as early life trauma, diet, both negative and positive exposures, also exercise. Then we look at their effects on all these different epigenetic or non-genomic mechanisms, and finally, how they lead to these neuropsychiatric disorders across the lifespan. This is me, as you would see, and this is how we started from, because this introductory video from a couple of years ago, now we have like a few more lab members. So to sum it up, we are interested in the susceptibility to neuropsychiatric disorders, and we think that they depend on our dietary or metabolic exposures, but also the emotional or psychological exposures in our parents. So if we just focus on how these different exposures lead to the neuropsychiatric disease risk in the same generation, that is something which I'll cover in the first part of my talk. And then in the second one, we'll focus on their effects, their non-genomic effects at the germline level, and how these susceptibilities lead to neuropsychiatric disorders in the next generation. So starting with the exposures in the same generation, our point of focus over here are these neurodegenerative disorders. So neurodegenerative disorders are very complex, but they are unified by some hallmarks, which is aggregation, pathological aggregation of these proteins that can be intracellular or extracellular. And there is this brain atrophy. Usually it is site-specific in the beginning, and then it becomes global. Now, I'm going to throw in a question there. If I ask you, which one of these guys do you think is more susceptible to neurodegenerative disorders, what will be your answer? Let's make it a little interactive, yeah. So if you can do a raise of hands, who thinks that this person is more susceptible to neurodegenerative disorders? This guy. Okay. Who thinks this person is more susceptible? Okay. Well, the situation is a lot more complex because we actually found out, as Vitek was saying, some very anti-dogmatic findings which were, well, I don't want to say game changers because then I sound like a show-off, but they definitely were very disruptive in what is usually thought about the link between metabolic disorders and neurodegeneration. Because we actually found out that in some cases, having a metabolic profile like this can actually be protective. And the story goes back to, like, my clinical years. This is, like, about 15 years ago when I used to have this hideous hairstyle, and I was working at Baylor College of Medicine. And there we were studying these two disorders, ALS, which is the most common motor neuron disease. And, it is, like, practically speaking, a disease with very high mortality. The usual survival is about 3 to 5 years after diagnosis, and the approved treatments, Riluzole and Edaravone, they have a median survival benefit of only 3 to 6 months. And the other disease is this frontotemporal lobar degeneration, FTLD, or what we call as frontotemporal dementia, FTD, when we talk clinically about it, which is the second most common cause of dementia after Alzheimer's disease and has no approved treatment. Now, interestingly, ALS and FTD are very much linked to each other clinically because a lot of patients who have ALS, which is typically like, things related to motor neurons, like, for example, rigidity, muscle atrophy, inability to walk, inability to swallow, like upper and lower limb symptoms, manifestations. They also start showing this cognitive impairment, which is frontal or temporal in its flavor, in its characteristic. On the other hand, a lot of patients with FTLD start showing these motor neuronal impairments and motor symptoms, which are reflective of ALS. The overlap is not only clinical, because actually one of my past collaborators, Manuela Neumann, she showed back in 2006 that there is also a molecular overlap between these disorders, which is related to pathological aggregation of TDP43 protein. However, there is one more overlap which we discovered and we have been studying over the last 12 years or so, and that is metabolism, where we showed that having this kind of metabolic profile can actually be protective in both ALS and FTD. So back at Baylor College of Medicine, I had access to the largest ALS clinical database in the world. At that time, like around 3,000 patients. And there we started looking at different, risk factors, demographic and epidemiological risk factors, and if they alter the disease course. So here you can see that we are comparing the age of onset, rate of disease progression and survival between males and females, between different ethnicities, and also importantly, between those who had pre-morbid diabetes, so who had diabetes before they developed ALS, versus those who didn't have diabetes before they developed ALS. And we actually thought that just like everything else, just like diabetes increases the risk for stroke, like in some cases also Alzheimer's disease, it would actually turn out to be a risk factor. But no, what we found out was that actually the age of onset in those patients who had diabetes was 4 years later. So diabetes was actually delaying the onset of ALS by around 4 years. Similarly, there was a non-significant trend towards increased survival in those cases with diabetes. So this was a finding which was, let's say, very anti-dogmatic, and we had to be very careful about it. And if you look at the title of the paper, it is also very sort of toned down, ALS disease onset may occur later in patients with pre-morbid diabetes mellitus. Now, the reason why we wanted to be careful was because there are so many confounding factors. We thought it could be maybe the anti-diabetic drugs, that diabetes patients take, which somehow are modifying the progression or modifying the pathogenesis of ALS. So then we looked at another group in which they are not taking any drugs, and the only differences could be related to dietary and metabolic factors. So here we looked at patients with ALS and we stratified them according to those who are gaining weight after disease onset versus those who are losing weight after disease onset. And we found out this is a visual representation of 30 patients with the highest BMI loss. And you would see that this is their disease progression level. And these are the ALS patients with the highest BMI gain. And you see how much more steeper the disease progression is among those who have this highest BMI loss. So weight gain, similar to how diabetes was protective, now we found out that weight gain is also somehow protective. And several years later, I ended up in Zurich. I had almost finished my MD Ph.D. when I collaborated with the group of Florence Rajendran and Rosa Paolocci was the first author, and we still continue to collaborate. We actually made another sort of very interesting discovery that somehow seems that the patients who have Alzheimer's disease and the patients who have ALS and FTLD, they don't really overlap. So if you have ALS, you are somewhat protected against Alzheimer's disease. So if you see over here, these are the autopsy specimens. These are just age-matched controls stratified across two groups, those before the age of 75 and those which are 75 years or older. Then we have ALS, FTLD, and here we have garden variety Alzheimer's disease. And we are looking at the level of amyloid beta plaques in the brain. And what you see over here is that, as we get older, even when there is no clinically diagnosed or clinically apparent Alzheimer's disease, still the beta amyloids deposit, they increase. This is just the normal controls without any neurodegenerative disorders, and you see that the age above 75, there's a huge difference. Of course, if there's Alzheimer's disease, the level of amyloid plaques is much higher. However, in those patients who have ALS, FTLD, they even have lesser increase compared to the age-matched controls. So somehow, ALS and FTLD are protective in Alzheimer's disease by reducing the beta amyloid plaques. And through some mechanistic studies, we found out that this is because the microglia in these cases, they were more active and they were more efficient in removing amyloid beta. And this is the last clinical study, which I will mention, and then we move on to more translational, preclinical, and basic science data. Here you are looking at nutrient signaling pathways and their role in amyloid clearance. So, we assess cognitive decline and dementia patients to this mini-mental score, which we list over here. And every year there's a decline of this MMSE score. Again, we have stratified the patients into two groups, above 75 and then 65 to 75. And what you would see over here is that there is a very interesting biphasic effect, metabolic disorders, on this clinical progression of Alzheimer's. If you look at the patients for the age of 65, you see that type 2 diabetes as well as hyperlipidemia, they're associated with the fastest progression of the fastest decline of MMSE. But if you look at older patients, again, we found out that hyperlipidemia turns out to be somewhat protective. So what you can gather from this is that these metabolic disorders in middle age are risk factors, but later on, they are turning into some protective factors. Now let's sum it all together. We found out that weight changes predict survival and rate of progression in ALS. We showed that diabetes can delay ALS onset and increase survival. ALS can be protective in Alzheimer's disease, and that effect is through microglial TDP-43 depletion. So microglia, when they have less TDP-43, they remove beta amyloid more efficiently. And finally, that metabolic disorders in old age are protective in Alzheimer's disease. Now, if we sum it all up at the molecular level, there seems to be a very interesting, I always call it love-hate triangle between TDP-43, microglia, and amyloid beta. And it seems like that this whole triangle, it is orchestrated by metabolism. So we have TDP-43 over here, a protein which is primarily in the nucleus. It gets pathologically misplaced to the cytoplasm, gets aggregated, leads to neuronal death. And we see that it has an effect which can be modified, or it has the disease caused by TDP-43, which is ALS and FTLD, can be modified by all these metabolism-related factors. On the other hand, we have this other part of the story where microglia are removing beta amyloid, and somehow they seem to have an effect with TDP-43. Now, what we don't know is if that effect is mediated through metabolism. So this is the starting point of these projects by my Al-Gabada Mosi and my postdoc Isabella Lepage-Raba, who are now trying to study these stories further and finding out what are the main characters involved. In this microglia amyloid project, our aim is to harness the power of microglia to preferentially remove amyloid beta without substantially harming healthy neurons. And because we have sufficient clinical clues, we think that this is achievable by targeted metabolic manipulations and interventions. So we know that microglia can be protective in Alzheimer's disease if they would remove this beta amyloid, which will lead to reduced AD pathology. But we don't want that these activated microglia cause neuroinflammation or they remove healthy synapses, process which we do know occurs in late stages of Alzheimer's disease. We want them to be preferentially phagocytosing only neurons. So to achieve that, we started with these HMC3. These are human microglia-like cells. We performed high-throughput metabolic manipulations, and then we exposed these microglia to amyloid beta and studied the uptake and degradation of amyloid beta through these very elegant assays, which I will explain in a minute. And at the same time, we are looking at microglial gene expression and doing all these functional analysis to see which pathways, which molecular cascades are relevant to this effect. So here you see some of the key results from this high-throughput metabolic manipulation. And you would see that we tested different conditions, varying levels of glucose, varying levels of polyunsaturated fatty acids. And essentially what we found out was that there was a bidirectional effect of lipids on amyloid beta uptake by the microglia. And when we removed the lipids from the medium, from the serum which these microglia had, so when we did this lipid starvation, we saw that there was an enhanced uptake of amyloid. On the other hand, when we supplemented either lipoproteins, like density lipoproteins, or we gave a non-specific lipid concentrate, we saw a significant reduction in phagocytosis. So the key word here is this bidirectional effect. Then we wanted to see what happens to this beta amyloid, which is taken up by microglia. There we designed an assay where we look at the amyloid beta, which is taken up by microglia, and then over 24 hours, we see how much of that is reduced. And what you would see over here is something quite interesting. So you see this is lipid starvation, increases the uptake of beta amyloid, which are these blue bars. Serum starvation, which is a general nutrient depletion, so not only lipids, but also proteins, etc., everything else. That also leads to an increased uptake of beta amyloid. However, when we look at how much of the amyloid remains after 24 hours, you would see that there is efficient degradation in cases with lipid starvation, but not with serum starvation. So both serum starvation and lipid starvation enhance uptake of beta amyloid, but it gets degraded efficiently only with lipid starvation, but not with serum starvation. So we were wondering why this happens. So then we looked at the pathway of, the fate of beta amyloid once it's taken up by microglia. So it gets into this early endosome, then this late endosome. Eventually, they combine with the phagosome to form this phagolysosome, and amyloid beta gets taken. And there are certain proteins which are responsible for formation of this phagolysosome. And what we found out was that in serum starvation, there is a slight reduction in these components, which potentially could explain why with serum starvation we don't see efficient degradation, while we do see it with lipid starvation. And mainly what we wanted to find out, what are the pathways responsible for these effects? And here you would see the heat maps for we did a sequencing which is both small as well as long RNAs. And we found that there is a clear differential regulation between these conditions. Here, this is the control serum. You would see that there are a number potential targets which are upregulated, a number which are downregulated. You would see that lipid and serum starvation, they have more or less the same pattern. However, you see that there is definitely an enhanced upregulation certain targets with lipid starvation compared to serum, and an enhanced downregulation. This part is more bluer, this part is more pinkish compared to serum starvation. And then we have identified now 3 targets which we are currently manipulating to see if they are responsible for the effects of lipid starvation eventually on this increased uptake and degradation of beta. And this is the pathway analysis. Interestingly, everything we studied and repeated multiple times, it all leads to this association with cholesterol biosynthesis, or this STREBF-based gene activation, or STREBF is also something which is very closely related to lipid metabolism, as many of you would know. Interestingly, we also found out that there are some pathways which are relevant to the SARS-CoV infections. This is kind of just a side observation we made. But then, there were a lot of epidemiological evidence about how people who have dyslipidemia, they are doing much worse during COVID. Perhaps this is somewhat relevant to that. The most important thing about these targets which we identified, PCSK9, LDL receptor, HMBS1, that these are all druggable targets. There are existing drugs for each one of these targets, which mean that we potentially can go into a preclinical trial and eventually, who knows, maybe to clinical trial and actually test all of our therapies. So the next challenges which we are facing, or the next targets for us, is to show that there is a substrate specificity of this effect. So we want to repeat our experiments not only with beta amyloid, but also neurons with tau pathology, neurons with TDP-43 pathology, apoptotic neurons, and healthy neurons. And there we want to selectively pinpoint the manipulation, which will preferentially remove either these neurons which are diseased, for example, with tau pathology or TDP-43 pathology, or the extracellular deposits of beta amyloid, but without harming these healthy neurons. We actually have some very promising data for that. If someone wants to talk about it, I'm happy to share, but I didn't put it here because it's the first experiment and it needs. Then, in collaboration with Professor Bożena Kaminska at Ninsky, we want to do a targeted manipulation. So we want to identify the pathway for which we have some candidates now. We want to manipulate that specifically in microglia, and then we will show that there is a functional impairment or functional enhancement of phagocytosis based on this target which we have identified. So this will be an in vivo validation of our work. Finally, I have a very strong clinical collaboration with Professor Paul Schultz at UT Houston, where we have already started a study in which we are taking serum and CSF patients, CSF from patients with Alzheimer's disease, and then we are performing these amyloid beta phagocytosis assays. We are segregating the patients according to those with high phagocytosis, those with medium level phagocytosis, and those with the lowest level. And then we are doing unbiased metabolomics and lipid profiling, segregate those. Again, there is some interesting data, but it is an ongoing study, so I'm not putting it in the slides, but I'm happy to share. So this is the very first interim key finding which I want to share. But we have shown that lipid starvation can enhance phagocytosis of amyloid beta by the microglia. Now, I talked about this love-hate triangle, right? And TDP-43 also was playing a role. And previously, we showed that TDP-43 depletion was enhancing the phagocytosis of amyloid beta. What's happening there? This is the other project about TDP-43, where our approach is to look at TDP-43 loss of function through these knockdown approaches, as well as study the TDP-43 disease-linked mutations. Do a very comprehensive analysis of transcriptomics, metabolomics, as well as these functional assays, which are in the next few minutes. Identify the target, do a CRISPR-Cas-based manipulation of the target and iPSCs, and make these brain organoid models where we show how the pathology can be, let's say how it's modified. So this is just an experiment which my student has performed, I would say 200 times now, always this TDP knockdown, always working very efficiently, followed by all these different analyses, functional, metabolic flux. This figure actually sums up a year of his work, where we found out that TDP-43 loss of function increase for glucose metabolism at all levels. Now, this is something very, again, anti-dogmatic and phenomenal. So what we found out was that TDP-43 loss of function, it leads to an upregulation of the receptor in these motor neuron-like cells, leads to enhanced uptake of glucose. Then we have upregulation of these key rate-limiting enzymes in glycolysis, as well as PCA cycle. And eventually, we also see an increased production of ROS as well as ATP. So through these functional assays, as well as transcriptomics, we showed that at all different levels, glycolysis, PCA cycle, oxidative phosphorylation in mitochondria, TDP-43 loss of function just enhances glucose metabolism. Now, the most important thing was to confirm it through this metabolic flux analysis. So here we have the data from seahorse. And I will just very briefly explain this assay. So what we try to do is that we block different components of electron transport chain. This seahorse analysis look at the effects at the exercise acidification rate as well as OCAR, and that gives us an indication of the functionality of mitochondria. And what we found out was that there is indeed an increase in ATP production. There is almost significant increase in respiration, their respiratory capacity is not mitochondria. So what seems to be the case is that it is clear that this mitochondrial respiration phosphorylation is enhanced on TDP-43 loss. This experiment has been repeated 5 times. Every time we get more or less. Then here we performed also an analysis, a metabolic flux analysis of glycolysis, the very first step in glucose metabolism. And here what we found out was that, again, with TDP-43 knockdown, there is an overall increase in glycolysis as well as the glycolytic capacity. Putting it all together, what we have with TDP-43 loss of function is a unique combination of what we see in cancer cells and what we see in some other neurodegenerative disorders. So usually cancer cells have this increase in aerobic glycolysis, what we call as Warburg phenomena, where they preferentially enhance their glycolysis. That ends up in increased exaggerated consumption of glucose, leads to all these kind of symptoms, in patients with cancer where they get cachexia and enhanced weight loss because all their glucose is up by the. On the other hand, typically in neurodegenerative disorders, we see an increased production of ROS, which means that there is an enhancement of oxidative phosphorylation eventually leads to oxidative damage and neuronal death. With TDP-43 loss of function, enhancement, glycolysis, increased glucose consumption, increased aerobic glycolysis, increased oxidative phosphorylation. Now that prompted us to think that what's going on, and actually this was the part where both me and Ismail, we got really stuck about what to do next, because we were actually hoping for something like this, not like this, you know. But the more we thought about it, the more, like, you know, it started becoming clear to us that it seems to be a defect cellular energy sensing. Cellular energy sensing is mainly the function of AMP kinase in the cells and in the body as well. AMP kinase, as the name implies, it is dependent on the levels of AMP. AMP is like, you know, sort of, you could say, a metabolite of ATP. So ATP is the currency of energy in the cells, as we all know. Once ATP reserves get depleted, we see more ADP. Once ADP reserves get depleted, we see more AMP. So AMP is a signal for the cell to think that, okay, energy is depleting. And then AMP kinase, it gets activated, gets phosphorylated, and then it leads to an enhancement of all these different steps, these different segments of glucose metabolism. And that is exactly what we see with TDP-43 loss. However, the crazy part is that here you would see like control cells, the blue ones, and you see that AMPK activity, it fluctuates depending on whether there is low glucose medium, normal glucose medium, high glucose medium. Now what we see with TDP-43 knockdown is that although there is a fluctuation, it's always much higher than what we see without TDP-43. So it seems to be the case where AMP kinase seems to be constitutively active, irrespective of whether there is enough ATP produced by the cell or not. So we wanted to also study it at the expression level. So here you have TDP-43, normal controls knockdown, that the knockdown is even more efficient at hours. Here we are looking at the level of AMPK and to like a mass fluctuation, but you do see a fluctuation phospho-AMP. And TDP-43 is knocked down, we see increased phospho-AMPK here as well as over here. And phospho-AMPK is actually the activated form. So again, this is like a validation of what we observed over here, that there is enhanced activation of AMP. Phospho-ACC is a downstream target of AMPK. So if AMPK is phosphorylated and active, then we see increased phosphorylation of ACC because AMPK itself is a kinase as well. And this is also something which here, hence proving that this increase in AMPK after TDP-43 loss of function, it has like downstream function. So to sum it all together, we have found out that indeed there seems to be this love-hate triangle between TDP-43, microglia, and beta amyloid, where we see that lipid starvation enhances phagocytosis of amyloid beta, and we see that TDP-43 loss of function induces a state of hypermetabolism in the cell via increased AMPK. Now, try to link both of these together. When there is the state of hypermetabolism, it means that the lipid reserves would be used up, meaning we are actually inducing a state of lipid starvation. And remember this neuron paper from 2017, where we showed that ALS and FTD, which have this TDP-43 loss of function pathology, they are protective in AD, which has this amyloid beta pathology. Perhaps this is where this. And just like some final words, so this is kind of ongoing work. We don't have this organoid-based model yet of ALS or FTLD or Alzheimer's disease, but we have made enough progress, and now we have these organoids. This slide is actually from like many months ago. Now we have actually organoids which can survive up to around 300 days, and they do show as well as now what we are trying to do is that we are trying to incorporate microglia with organoids. It's an ambitious project, but if we succeed, then we will be able to validate all our findings. So that's the first part of the presentation, and I would suggest that maybe we do the Q&A for this now, and then we can go on to the. Thank you very much.
Thank you very much, Ali. Very interesting, thought-provoking lecture. Do we have questions from the audience? So if not at this moment, let me go with my. This lipid starvation, do you think that the effect has very specific to media and amyloid, or in general stimulator of endocytosis or phagocytosis, other cells?
No, that's a very good question. So we haven't done this experiment ourselves, but there is like similar data in macrophages that actually the macrophage-induced phagocytosis is also enhanced with lipids. Now the tricky part is that we don't know whether there is any substrate specificity, because usually these macrophage experiments, they were done on beads, and what we are looking at are more physiological targets, like healthy neurons and pathological targets.
Question from the audience. And in ALS studies, question or outcome, BMI changes are included in the metabolic activity.
Again, a very, very pertinent question, and that's why we started looking at the changes, in BMI after, like, within the first year after diagnosis. Because in late stages, you're absolutely right that there's too much muscle wasting. Also, in late stages of the disease, patients lose weight because they have inability to swallow, so they have to be shifted to this nasogastric feeding, and that is why we only looked at the initial part of the disease. However, again, I think the clinical evidence for this was just, suggestive. But now that we have clear molecular data, I think it's becoming more and more clear that these metabolic pathways are integral to pathogenesis.
Thank you. Last call for questions from the audience.
Hi. I have a question regarding your life. So the fact that you are establishing brain organoids to study properties of function during brain development, how do you correlate metabolism during development and then essentially the establishment of neurodegeneration?
Yeah, so it's a bit ambitious because as many of you would know, that organoids are a good model for development, but not exactly for neurodegeneration. However, what we want to harness over here is this possibility that we can edit, already the IPS lines, So what we want to do is that these candidates which we have identified, we, CRISPR-Cas added them during the IPS line. So when we would make the organoid, already that part would be it's come somewhat like a knockout model, in terms of let's say seeing whether neurodegeneration will appear or not. There is evidence actually now also from patients with Alzheimer's disease that if we make organoids from these, we take fibroblasts from Alzheimer's patients, we reprogram to iPSCs, then grow organoids for them, we do see that they start showing this tau pathology at some point in time. With amyloid beta, it's tricky. A lot of times it has to be induced. But definitely this model has the power that we will be able to look at everything like, sequentially, plus we can edit the IPS line. There are certain metabolic, the peculiarities to how like stem cells are behaving. And we would have to, let's say, take them into account because IPS, they have their own metabolic adaptations. These are certain challenges in this model. So that's why a combination of human cohort studies, in vivo investigations, animal models, as well as organoids, probably all are needed to reach the right answer.
Can you tell us a bit more about the TDP functions? So I assume that now it's like new, but what's already described about TDP? Is it like common for all types of cells, only brain specific?
Yeah, so TDP43 is quite ubiquitously expressed. It's practically present in all different tissues, all different organs, and it has like a multitude of functions. It is important for regulation of alternative splicing, in microRNA biogenesis, in RNA metabolism. These are some of like the key roles for it. As long as TDP43 is nuclear, it is functional. Once it gets into the cytoplasm and gets like aggregated, then we think that mostly there could be a toxic gain of function because sometimes the TDP43 deposits could be formed on microglia and then they disrupt mitochondria and then they disrupt mitochondrial functions, but there is no specific physiological function of cytoplasmic TDP43.
Thank you very much. Last opportunity.
Hi, have you maybe checked if the lipid starvation affects somehow the cell membrane composition, which could contribute to the changes in endocytosis, etc.?
We haven't, but we'll do now.
Thanks.
Thank you. I have a similar question because I would like to ask about the lipids, for example, HDL starvation on the effect of beta amyloid metabolism. Do you know if it is the effect of, for example, HDL starvation itself, or maybe the altered action of HDL binding proteins in lack of HDL?
Yeah, excellent question, actually. So the effect seems to be, I would say, like, it's a lipid-associated effect. We don't only see the effect with HDL, but also with lipid concentrate, with like, a combination of HDL, LDL. They all have this bidirectional effect. And actually, like, in the beginning, I was also very much interested if it has something to do with the downstream signaling. So we did like an experiment where we knocked down HDL receptor, SRB1, the scavenger receptor B1, and we didn't see an effect. So it seems to be more something which is like, nutritional at the metabolic level rather than dependent on some downstream receptor.
That do you think, because really gut micro changes, right? It could also have some.
Again, it's an excellent question because also, as far as like my knowledge goes, I think at least half of the metabolites we have are coming from our microbiome. So they could be playing a key role in this. And then we also know that the microbiome gets altered, in diabetes and like other metabolic disorders. It's not my expertise. I do know, like, that there is some important work going on by Anthony Hannon in Australia, one of our collaborators, about this. And Jamie Hackett in EMBL Rome. You can definitely follow their work. I don't think anything is concretely published as of yet, but definitely they are looking at microbiome and metabolome released from microbiome and how important that is for neurodegeneration. Very good question.
Okay, thank you very much. I guess if you have more questions, maybe there'll be time after the second part of the talk.
Sure, yeah.
Thank you.
I'll just take a sip of water and then I will move on the second part. And actually, it's not that I prefer like one part of my work. There is one part of the work which witnessed, and that is very mechanistic about pathways. And then there is this side of our lab, and we are doing work which involves a lot of different parts and has a lot of implications, humanitarian causes and compassionate causes. Like I believe in a combination of prosody and compassion. This is very fitting to work on both these topics. Here, I want to look at the metabolism, insusceptibility, how they affect pathology, the I was compelled to modify it and make it. Again, it seems that metabolism. Think about whether parental effects all inheritance and many different and I comprehend together with these friends of genetics a couple of where we looked at all potential evidence is inheritance. As well as potentially altering the risk of metabolic disorders. Intentional. Involvement of hammered by it and stay care and different kind of like horrible interventions with the reason why we do that is because the very repetitive lot of different biggest risk factors and adulthood psychiatry actually parent childhood abuse. But in an unpredictable fashion every day. Combination very circuitry and brain neurotransmitters causing last generation. Started looking at genomic analysis, we took wild type mice. Psychological, because we were able to lead the germ cells, brought us to the next question, that what is inducing these changes in the germline? And for that, we performed an experiment where we did a blood transfusion. So we took the serum from the mice after trauma, injected those into wild type mice to see if this trauma could be transmitted. And then we assessed the germ cells as well as the next generation. And indeed, we saw that there was this transmission of trauma just through injections of these serum. So blood was transmitting trauma from one, the affected mouse to the unaffected mouse, converting a non-traumatized mouse into a traumatized mouse in lay terminology, and leading to this intergenerational phenotype. But the question which still remain unaddressed is that, okay, it is the serum, but serum contains a lot of different factors. Serum contains non-coding RNA, serum contains cytokines, serum contains other immune factors, hormones. So what is responsible for this transmission? And is there a mechanistic hub which causes these germline changes, which are then passed on to the next generation? The other question which remain unaddressed is that how long do these changes persist during embryonic development? But actually, if we talk about the field of epigenetic inheritance, the challenges do not end here. Perhaps the biggest challenge is the challenge of translation. Because when we induce trauma in this mouse model, there are questions about whether they are similar to the complexity of trauma in human conditions, whether there's face validity of this model. What about the transmission which could take place through body fluids? And it's not only the germline which could be responsible, right? I mean, think about a mother who is traumatized and then is lactating and potentially passing on these trauma-induced factors to the baby. What about those? What about biological versus psychosocial transmission? If we apply it to humans who get traumatized, they also could have impaired parenting styles. There could be family dysfunction. There could be a lot of these factors through which trauma could be transmitted, and they're all fundamentally important. What about confounding influences? Genetic heterogeneity. We study everything, in mice which are inbred. They are genetically very homogeneous. When we think about human conditions, there are ethnicities, there are all these different factors, and we are genetically a lot more heterogeneous compared to mice. The germline assessments are very challenging. Of course, there is very limited access to, sperm and seminal fluid in males. Definitely for oocytes, you actually need surgical procedures, so that access is practically non-existent. Finally, what about the mechanistic and causal underpinnings? Can we do a combination of human assessments with the assessment in our experimental models to show that there is mechanistic relevance of this? So for that, we are using a very, I would say, unprecedented combination where we have a mouse model, we have 3 different human cohorts where we do have access to the germline, and then we are also studying the effects at the organoid level. So what we did in the translational approach is that we identified 3 different cohorts with varying levels of genetic heterogeneity. Now, what you see over here is a world map, but it's a very interesting world map because it is the level of consanguinity, cousin marriages, which happen in different communities across the globe. Now, consanguinity leads to genetic homogeneity. Consanguinity is basically inbreeding, which we do in mice. So we selected 3 different cohorts. The first one actually is from Pakistan, which actually happens to be the country with the highest level of consanguinity in the world. I mean, practically everyone in Pakistan is like first or second cousin of each other, And we collaborated with the SOS Children's Village, where we identified a cohort of children with paternal loss and maternal separation. So these were children who were, who unfortunately lost their fathers within the preceding 1 year. And because the mothers, they were destitute, they were not financially viable, they had to give up the children for adoption. So there was this very tragic scenario of forced maternal separation as well as paternal demise. And then we identified another cohort of adult men who were exposed to either simple or complex childhood trauma, where we also analyzed the sperm sample. We now have ongoing study in this cohort in Bosnia, which happens to have mid-level of consanguinity. And there is this, verified exposure to trauma because there was this very tragic event of Srebrenica massacre back in 1995. A lot of children who were exposed to this massacre, they themselves have children now. And here we are able to assess whole families in which serum and sperm from fathers, as well as serum from children. We actually also have serum and other assessments from mothers, but specifically we have serum line fathers who were children at the time of this genocide. Finally, we have a third cohort, which is actually based in Warsaw, where we are studying mother-child dyads, where there was a history of childhood trauma in the mothers. And this is the assessment from this cohort in Pakistan, where we studied these PLMS versus control children. And again, these heat maps indicate that there definitely seems to be a clustering of certain small RNAs. There are certain small RNAs which are, downregulated in this red group, which is the PLMS, the children with trauma. And there are some others which seem to be upregulated. But when we go into the effects of trauma in the sperm, it seems to be a more consistent pattern that we see that a number of these small RNAs seem to be upregulated, like, in adult men with complex childhood trauma. This assessment was done in the sperm level. Now this becomes more and more interesting. Yeah, like now we are looking at heat maps, but things start to become like very mechanistically relevant and targeted. But first I want to show you how do we account for psychosocial trauma. This is the Action Bosnia cohort. So there we are recruiting these families. There are certain pre-procedures which are followed, which is overnight fasting so that no metabolic parameters are affected. Standardized breakfast for children. Fasting is for parents. Then, of course, some procedures which they have to follow before depositing the sperm or seminal fluid sample. Semen is collected from fathers, sperm is pelleted, and seminal fluid is separated. Similarly, there's blood sampling from all adults as well as children. And then a very comprehensive psychometric assessment where we look at not only childhood trauma questionnaire, but PTSD screening, parenting scale, family functioning scale, childhood trauma screen, children behavioral functional scale, scale for anxiety-related disorders in children, scale for epidemiological studies for depression in children. So we are doing a very, very comprehensive assessment and a lot of different lab parameters. Similarly, we are trying to account, partially at least, for the body fluid influences in this cohort in Wrocław. Now here, as I mentioned, we have these mother-child dyads, and we have access to the human milk samples from these mothers, which was collected between, the first 2 years, right after firstborn child. Now those children are between the age of 4 to 5 years, and we are doing psychometric assessments as well as blood sampling and serum assessment and several laboratory parameters. a lot of this data has already been analyzed. So I already showed you the effects of trauma at the serum level in children and in the sperm of adult men. And here there is RNA sequencing data from these human milk samples. And as like my PhD student, she always says, like, some of this data is too good to be true. And actually this seemed to be the case because we found one particular microRNA, mu223-3p, which is similarly altered. Now these are three different cohorts and three different specimens. We have serum, sperm, milk, different age groups, just after childhood trauma, but three different cohorts. And we see mu223-3p to be upregulated in sperm, in serum, and in milk after trauma. Interestingly, all the microRNAs which we were able to validate, they seem to be very strongly linked to lipid metabolism as well, which has brought us to this hypothesis, which we are currently validating, that lipid metabolism seems to be the key mechanistic hub. Now, lipid metabolism definitely could be important for this transmission of epigenetic factors of all these different non-coding RNAs from the periphery to the germ cells, or from the periphery even to the brain, because lipids can cross barriers. They have access practically to all different organs in the body. When we look at all these different factors which have been associated with exposure-based inheritance, think about how many of them are also related to lipids. Here you see exercise alters lipids. High-fat diet alters lipids. Aging, as we grow older, we tend to get more dyslipidemia. Psychological trauma, we already showed that lipids can be affected. And actually, when we look at our own data, now this is data from MSUS mice, where we see a reduction in HDL, high-density lipoproteins. This is the data from these PLMS children, where we see changes in two different parameters. We see a reduction in HDLs, as well as an increase in triglycerides. So there is enough evidence that lipids could be crucial to this transmission of trauma across generations. And indeed, one important aspect to consider is that there could be a strong partnership between these small RNAs, which we already showed that they are altered, as well as lipid metabolism, because lipid metabolism is crucial for the release and circulation of these non-coding RNAs. So the non-coding RNAs, they could be released in 4 different forms. Either they are packaged as these extracellular vesicles and released in the circulation, or they can be incorporated into HDL, high-density lipoproteins, and released. They can also be released when a cell undergoes apoptosis. And fourth, they are released as these protein complexes with Argonaute proteins. 3 of these are associated to lipids. EVs can cross, like, the lipid bilayer membrane. HDLs are lipoproteins. Apoptotic bodies are not lipoproteins, but they are taken up by the recipient cell through the same receptor which the HDLs use. So SRB1 is responsible not only for uptake of HDLs, but also apoptotic bodies. So again, lipids seem to have a very strong partnership with these circulating non-coding RNAs. And this is what we are doing right now, that we are extracting HDLs, EVs, and apoptotic bodies after this MSUS model, and we are studying them at, I would say, a very comprehensive molecular level. And then we developed this cross-injection strategy model. So we took 2 different exposures. So here, this is MSUS, for which we do know that there is intergenerational transmission of trauma. And we incorporated 2 exposures which we do on top of MSUS. One is exercise, so something which modulates lipids in a good way, and high-fat diet, which modulates lipids, in a bad way. And then we want to see whether these effects, these intergenerational effects, would be altered. And we have some interesting results already from some metabolic results. We haven't done the behavior, but at the metabolic level, we have done some assessment in the offspring. Now, this is the offspring born from the MSUS mice. And you see that this is the glucose tolerance test where we give a bolus of glucose and we see how the body responds to it. It should come down at some point. And we see this impaired glucose tolerance in the offspring of mice born after the MSUS model and the parents. So trauma offspring has this impaired glucose tolerance. This is the offspring born after injections of high-fat diet. And overall, you see that it didn't reach significance, but we look at the initial response of what happens when you inject glucose. We again see something quite similar to what we see. So the effects of HFD on these initial points after in the glucose tolerance test are mirroring. They are very similar to that of the MSUS model. On the other hand, when we see this effect of exercise, we definitely see a partial recovery. We don't see this initial impaired tolerance. Now, this is just very new data. We need to, let's say, study, we need to do a comprehensive phenotyping, but it has given us a very important clue that perhaps these lipid modulating strategies could have effects which could also be transmitted. So not only, trauma could be transmitted, but perhaps trauma healing in the form of this exercise, environment enrichment, lipid modulating strategies could also be transmitted to the next generation. Then we want to perform this very important experiment for which actually we already have these very nice mice from VTech. We want to do this adipose tissue transplant. The idea here is that if we stop the release of microRNAs from the adipose tissue, then the adipose tissue would not be harmful anymore. And if we would do this adipose tissue transplant from mice in which this microRNA release is blocked because the dicer protein, which is important for microRNA biogenesis, is blocked, then we won't be able to transmit this trauma to the next generation. This is something which probably we'll need like another year or so to perform. Right now, I'm looking for actually any collaborators who can teach my student how to do adipose tissue transplant. So if you know someone, please let us know. And finally, we are also looking at the germline persistence of these effects. So this is in collaboration also with like a former student of VTech, with an expert in embryo transfer. So we are manipulating specifically these microRNAs in the germ cells, and we want to see how long they persist during embryonic development, what are their targets, and eventually after also a confirmation of offspring phenotypes. And lastly, I want to touch upon the implications of this research for the society, humanity. This is actually photo which a lot of you would have heard about or would have seen somewhere. This is known as the Afghan girl. She got featured on National Geographic back in the 80s. Became like a symbol for the plight of children, the tragedy which children face through whenever there is a conflict or genocide or war. Several years later, she was rediscovered. This time she had a child herself. And I don't know, like, it's of course very hard to prove biologically, but there are several reasons why she could have transmitted to her child. And if we just think about how relevant this work is to currently what we are facing, the war in Ukraine, which practically for me is, I was speaking to a journalist like a few days ago, and I made this statement, which again I kept on thinking about, that when you talk about the type of trauma which the Ukrainians are suffering right now, which is very complex in nature, and it is affecting potentially also their children. Like, with this MSUS model which we talked about, this is actually being implemented over. And at the same time, Ukraine is a community which has been repeatedly exposed to these like wars and genocides. There's already evidence that intergenerational trauma exists there. It has not been studied at the molecular level, at the epigenetic level, but we do see an increased risk for this stress reactivity. it's very important to think about how science could be applied to these scenarios which the humanity is facing. And also, if we can apply whatever we learned from science to help out these populations. And this is kind of like a mission for our lab. So apart from science, we also write like all these different features. We had 2 features last year in Nature Human Behavior and Nature about this. And there was some level of satisfaction that at least we could sensitize the community, the scientific community at a larger level, and perhaps even some policymakers about the implications of our research and their relevance to the world. Thank like all the collaborators who have been involved in this work. You see some of these names are coded a bit differently because we form part of this tremendous consortium. That's the name. It is this. We were recently awarded this EU P.H.D. grant. Then we also have collaborators. Definitely, I want to thank VTech for the mice and also for passing on Asha to us, who's very skillful. And it's been like a wild ride, as I would say, in the last 3 years, working with this team, which we have put together. A team of like very young, dynamic individuals have performed like, I would say, beyond my expectation, and more than brilliant. And this is how we look on a regular day. This is how we look when we get a grant. So thank you very much.
Thank you very much.